Thank you so much for the opportunity to speak here today. It's been really great to hear about this institute and the work that's being done here. I'm a probabilist, and I work in this area of phylogenetics, and I'm going to tell you a little bit about the sort of work that we've been doing. Let me just kind of jump right in and tell you what this talk is about. And so the basic problem that we want to solve is the following. I'm sure that many of you are familiar with it. We have a bunch of species, say these are Darwin's finches. These are Darwin's finches, and the way that they've evolved, just maybe the cartoon version of it is there was maybe an original sort of species on some island here in the Galapagos Island, and then some group of bird migrated to a different island, sort of got cut off reproductively from the first one, adapted to a different environment, formed its own species, and then the process repeated itself. Another group went to a different island, a subgroup went to another one, and so forth, until you have sort of the current sort of diversity of Darwin's finches that you observe today. And so a very natural way to represent this kind of evolutionary history is through a tree diagram like this, right? So where you have, this is called an evolutionary tree or a phylogeny. You have sort of at the root of the tree down here, this common sort of ancestor, and then each branching in the tree corresponds to a speciation event where a new kind of species got created, and the previous one sort of continued in the other direction. And then at the tip of the tree here, you have these sort of contemporary species, the kind of current species. And so the goal here is to reconstruct this evolutionary history in the sense of reconstructing this tree. This is called sort of phylogenetics. And the way that you do this is you get a bunch of genetic data from all of these species, and then from that you try to extract the sort of the tree structure that's underlying this evolutionary process. So biologists have been doing this for a very long time. There's lots of methods for doing this. But there's also continually some new sort of challenges and opportunity. In particular, nowadays with the sort of the growing availability of sort of genomic scale data for lots and lots and lots of species, there's an opportunity in the sense of being able to extract more information about the evolutionary past. But at the same time, of course, there's challenges in analyzing this data. One obvious one is sort of the more kind of computational side of things, right? How do you process lots of sort of genomic scale data for thousands or millions of species? But I'm going to focus in particular on some of the more kind of modeling aspects of how do you model these sort of complex genomic evolution problems, and how do you infer from this, so the sort of statistical questions that are attached to this. So there's a lot of work being done on this these days, in particular in the sort of more mad bio community. We just finished this semester program at ICERM that brought together people from lots of different areas of mathematics, algebra, geometry, probability, combinatorics, with computer scientists and biologists and statisticians looking at some of these questions. So I'm going to give you a bit of a biased perspective on some of these challenges from the point of view of a probabilist who's interested in sort of proving theorems about some of these questions. So this is going to be part tutorial, part sort of survey, part new results. I'll start by telling you about some classical things about how you build trees from genetic data and the kind of methods that people use these days. And then I'll focus on sort of one, just for concreteness, sort of one question that you might ask about these methods to kind of drive some of the theory to develop new methods and understand what people do in practice. And then I'll tell you about some of the more technical aspects if I have time towards the end. Okay, so here's the, so let me start just by the basic object that we'll be interested in. So we'll be talking about trees. Throughout, I put some definitions here, but for those of you who are curious, but I'm not actually going to prove anything, so you don't actually need to read them. I'm mostly going to show you some pictures. Okay, so we're interested in trees. What is a tree? Just to make sure everybody is knows what I'm talking about here. A tree, of course, is a graph, so it's got vertices, which are these dots here, and then it's got edges, which are pairs of vertices, which are these lines connecting them. And a tree is a special kind of graph that's, first of all, connected, so you can go from anywhere to anywhere by following the edges. And it doesn't have any cycles. Okay, so that's what a tree is. I'm going to be, now what I want to emphasize in this picture is that I'm going to be interested in one very special kind of tree, which is that it's called an X tree or phylogenetic X tree, where I'm going to put labels on the leaves. Okay, so the leaves are the degree 1 vertices, the ones that have only one neighbor. And these are important because this is where the data lives. This is where my species are going to be. So think of 1 here as dog, and 2 as cat, and 3 as frog, and so forth. Okay, so what I'm interested in ultimately in reconstructing is this sort of tree-shaped history of their evolution. Sometimes I'm going to have a root. So it's very natural to have a root here because these are sort of processes in time, so they start somewhere. And that's sort of where the root is. But a lot of the statistical methods that I'll be talking about are not necessarily able to reconstruct the actual sort of origin of where this process started. So sometimes I'll be using unrooted trees or rooted trees, depending on what I'm talking about. Okay, so hopefully this won't be too confusing. And of course, I'm interested in these trees up to isomorphism. Right? So I don't care how you draw them, as long as it's the same graph, it's sort of the same tree shape. And again, because I care about the leaf labels, where the species are on this tree, these have to be, the position of these have to be the same. So here, human is next to mouse, and so forth. And so these are the same 2 X tree, if you want, or phylogenetic trees. And do stop me if I don't make sense. Now, so the tree represents this sort of sequence of speciation event, if you want. There's another natural way to think about it is that it also gives you a sense of how far apart things are. Right? So the further apart things are, the more kind of different they are in some sense. And so there's a natural sense here of thinking of a tree also as a metric. So here's what I'm going to do. I'm going to put on each edge of my X tree here, I'm going to put on each edge some positive weight. And then I'm going to think of this as a metric on the leaf labels. This is actually a more general X tree where there's also a label on one of the internal vertices, but just ignore that. I'm going to think of this as a metric on the leaf labels by saying, well, for any pair of leaves, there's a unique path between them. This is one property of trees. There's only one way to go from anywhere to anywhere. And so I can sum the weights on this path. And that gives me a notion of distance between these two species. Okay, so if you look at this table here, for every pair, I have the sum of the weights on each corresponding path. And so what these weights are going to be, you could think of it as they could be some amount of time, right? So it's going to be the time between these 2 speciation events, or it could be something more sort of genetic related. It could be sort of the expected number of mutations per site on that edge. Okay, so we'll come back to that in a second. Now, the one great thing about these metrics is that you can use them to reconstruct trees. It turns out that if I give you a tree like this with edge weights, it's easy to construct that metric out of it. But if I give you a metric that actually comes from one of those trees, there's sort of a unique tree and set of edge weights that you can get out of it. And here's just a little trick of how this is done. This is all sort of very classical things. Suppose that I have 4 species and I'm trying to reconstruct the tree on them. If you think about, one thing I should say also is that I will always assume that my internal vertices have degree 3. Okay, so these are sort of branching trees. I will suppose that I have 4 species. Then if you think about the trees that you can have on 4 species that are binary like this, there's really only 3 possibilities, which are these 3 over here. And this is really about whether A is sort of next to B, or A is sort of sister with C, or A is sister with D. Okay, so suppose that I'm trying to figure out which one it is, and what I have is one of those tree metrics. I have delta AB for every A, and delta CD, and so forth. And so it turns out that you can figure out which one it is by computing what's called a 4-point condition, which is this particular quantity there. Okay, so why does this work? Suppose that the truth comes from the first one. What's going to happen when you compute this particular quantity, you'll see I'm adding AB, adding CD, subtracting AC, subtracting AD. Because of the way that these tree metrics work, where they're the sum of the weights on the edges, you get all this sort of cancellation. This is going to cancel with this, this with this, this with this, and this with this. What you're going to end up with is something negative. So if the true tree is this one, from that quantity, I'm going to get something negative. And now if you try the same thing on the other ones, this one is going to give you something positive. Again, if you just do the cancellations, you'll see what happens. You get sort of twice the length of that middle edge at the end. Or if you do it on this one, you get 0. So in other words, if I give you a tree metric on 4 leaves, you can figure out just by seeing whether this is positive, negative, or equal to 0, which tree it actually came from, which leaf-labeled tree it actually came from. Now, this is what happens if you have 4 leaves. If you have more than 4 leaves, well, there's different ways of doing this, but you can think of this as sort of a little puzzle. You can do this for every 4 tuple of species, and then you get all these little pieces that you can try to put together into a full tree. It turns out to be relatively easy, computationally speaking, so I won't tell you how to do this, but it's sort of a straightforward process. Okay, so that gives me one possible way of reconstructing trees. Now, of course, in reality, so first of all, where am I going to get these distances? And second of all, if I have data, figuring out something is equal exactly to 0 is not going to work very well. So I have to, if I want to turn this into an actual method, I have to do something more. So where is this information going to come from? This is where the genetic data is going to come in. Okay, so the way that biologists reconstruct these trees is that they collect genetic data for all of these species. So they take one representative individual from each of them. And then in the simplest possible case, you might take just sort of one gene that they all have in common. This would be an example where you have all of these 12 species, and I'm giving you the sequence of this representative individual, the sequence of this particular gene in a representative individual for each of them. And of course, the basic idea of why this is useful is that if you take 2 species that are sort of close together in the tree of life, they will tend to have very similar sequences in the sense that there has been very little time between them to have sort of mutations or substitution. So there's going to be a few differences. Whereas if I take 2 species that are further apart in the tree, you're going to see a lot more differences. Okay, so somehow the genetic data allows you to get a sense for how far apart things are in the tree to begin with. And so how do I turn this into an actual distance is maybe less obvious than it seems at first. So this is where probabilistic models are going to start playing a role. The basic model here that I'm going to use is that these DNA sequences are going to be evolving along the tree of life by a process of single-site substitution in continuous time. So it's going to be a continuous time Markov process on a tree where in time, an A might become a G or a C might become a T. And so it's going to look something like this. Suppose this is my tree. If I look at one particular position in the DNA, right, there's going to be, the ancestor is going to have some value there, say A. I'm going to imagine that this was picked according to some stationary distribution of this process. And then along each branch of the tree, sort of independently, I'm going to start from the ancestor's position and then run this continuous time process that was going to run for a certain amount of time t at a certain rate mu. And in the simplest possible case, but you can generalize these, in the simple possible case, at this sort of rate mu, you have a mutation where your A becomes any of the other 3 possibility equally likely. And then this process just continues. And then you end up with a value for this descendant. And then you repeat the process independently over here and over there. And then at the end, you get a particular nucleotide for each of your current species. This is, just to simplify here, I'm showing you just sort of the first 3 rows of this table. I've got 3 species here. This could be the tree between them, and I'm running this continuous time Markov chain on them to kind of explain what the distribution of the data might look like. And so how do we get the distance out of this? One thing to note is that even though along this branch of the tree, I run this thing for time t3 and mutation rate mu3, so the expected number of substitution is going to be sort of linear in t3 times mu3, but I'm only observing the end process Right? This can go back and forth between different states, and so I have to take that into account. And so if you use Markov chain theory, you can write down a formula for the probability that if the ancestor had an A in that particular position, what is the probability that there's going to be a G in the descendant? And so this is what the formula looks like. It depends on T3 mu3, but through this more kind of nonlinear thing that kind of captures the fact that you're kind of converging to a stationary distribution there. Yes?
Assuming you've just observed every single species in the tree and that there's no missing species, or how is this working?
So I'm observing, no, so I don't observe every species. I'm really just observing the ones that are at the leaves of the tree, of my tree, right? And I'm just assuming that, that I'm just connecting them with the tree that's between them. And so in particular, if there's been some extinction or other things, they just don't appear in my data.
Okay, and so is it identifiable then?
It will be identifiable. I'll say that in a second.
Oh,
Yes.
okay, all right,
Yes.
thanks.
Yes.
And my question was, what if some particular, you said that each substitution is equally likely. What if some particular substitution is viable?
That's right. And so we're kind of sweeping under the rug that there's a lot of things going on here. In particular, selection could be playing a role. We're kind of just imagining that most of what's going on in this genetic data is just this sort of neutral, almost neutral evolution that I can model by this sort of very simple kind of continuous time Markov chain. There's going to be some of these sites that actually have selection going on, and the model will not really reflect that very well. But at the end of the day, what we care about is to get a sort of a sense of how far apart things are in the tree. As long as a lot of this is sort of neutral, this model will sort of capture a little bit the kind of the clock that's going on there without being too affected by this. But yes, the model is definitely not perfect. And of course, here I'm doing this Zoox counter model where you can jump equally likely between different states, but you can do a more general thing as well. Yes, I think there was a hand over there.
And are you selecting genes for which the traits you're interested in, or all the genes?
So if your goal is to reconstruct the tree, in fact, you may not even want to use coding things because of the selection. And so you might want to look at pseudogenes or other parts of the genome that have more kind of neutral sort of evolution going, so that you're really just capturing this sort of clock that's ticking in some sense. But in practice, biologists will just take whatever they have and run these models and see what they get. And then the genes will have different lengths. Usually, so it depends what kind of data you have. But these they will, yes. So if you could have thousands of nucleotides, or they could just be hundreds, depending on what technology they're using. And they'll use whatever they have for this. And so the model doesn't care about the length of the gene. I was going to say that in a second. What I'm showing you is just the evolution of one site. And then I'm going to imagine that each site evolves independently according to the same model. So each column in this data runs according to this continuous time Markov chain, and that gives me the distribution of each of these columns. Is there a question over there? Answer? Yes. The other thing that I'm sweeping under the rug, of course, is I'm only modeling substitutions when actually there's a lot of other things that are going on. There's insertions, there's deletions, there's rearrangements. And so I'm assuming that there's in a first kind of pre-processing step, I get this nice table where I've sort of aligned all of my nucleotides across species and kind of remove the gaps, and hopefully kind of got rid of sort of the insertions and deletions. And I'm only looking at the effect of sort of single site substitution. And so how do I get a distance out of this? Right? So I can look at a pair of species. I can count how many differences there are between them. That is this quantity over here in purple. So it's the sum of the differences, the counts of how many times they have something different. But now, because of this nonlinear transformation that I showed you on the previous slide, I basically have to do the inverse to get something that's sort of linear in t times mu, basically. And so from this, what you can show is that, okay, so here comes the theory. If I have a table like this with infinitely many columns, by my assumptions, this thing is sort of going to converge to sort of the true kind of distance between the species. And once I have the true distance, by the example that I showed you before with the four-part condition, I can actually recover the tree out of it. And so the identifiability of the tree actually comes from this previous argument that I gave you about the fact that if I give you one of those distances, you can actually reconstruct the topology from it. And then the model itself allows me to estimate one of those tree metrics, and therefore I get a unique way of reconstructing a tree out of it. So this is what's called statistical consistency. If I have an infinite amount of data, I'm sort of guaranteed with this particular method to reconstruct the true tree, assuming, of course, that this model is correct. So this is one way of doing this. In practice, what biologists often do instead is they use something called maximum likelihood, where once I have a model like this, I can write down the probability of observing a particular table given a particular tree and a particular set of parameters. So if T is my tree and theta is the set of parameters on the branches of the tree, then by independence and so forth, I get that the probability of observing this table is just this product of probabilities of each of them. And then I can choose the tree and the parameters that maximize the probability of observing the data. Right? So that's maximum likelihood. This also has nice properties. In particular, it's consistent, right? If I have an infinite number of columns here, it's guaranteed to converge to the right thing. The downsides of this is that, computationally speaking, it's much harder to do this optimization over the space of trees. There's a lot of trees and leaves in general. But in practice, there's some good heuristics for doing this that people use and are happy with. And so in practice, this tends to give better inferences, even though it might be a little slower to kind of converge. Okay, so that was the more classical stuff. Am I doing on time? And so where things are going to get a bit more complicated is, so far in my sort of story, I've sort of assumed that I've got one gene that I've sequenced across many different species, and I'm using that to reconstruct a tree. When you have a sort of genomic scale data for all of your species, then you may have many different genes or different regions of the genome that you're using for doing this reconstruction. And one challenge there is that it turns out that different genes are going to tell you different stories about the evolution of these species. So I have to distinguish between two different trees now. There's going to be what's called the species tree, which is going to be this, is always going to be these sort of fat-looking trees. This species tree is the thing that I'm trying to ultimately reconstruct, at least in this talk. This is what my goal is. And this is what I started with, right? This story about Darwin's finches sort of evolving by speciation. Each branching here corresponds to a speciation event in that history. A gene tree is going to be the history of a particular gene. And as you can see on this, so these are going to be the thin trees, and they can have a different sort of shape than the species trees in general. And there's lots of reasons why this is true. There's lots of phenomena that lead to different genes having different sort of histories. One of the more obvious one is horizontal gene transfers. If I have a gene that sort of transfers between two unrelated species, it sort of suddenly and replaces an existing gene. It creates a different shape of the tree. Another thing that creates this sort of thing, which is less obvious, is gene duplication and loss. It can create the impression that certain genes have a different story. These are important, but I'm not going to go through them here. The one that I'm going to focus on more is the one that the people in phylogenetic literature have been really interested in. This is called incomplete lineage sorting, and I'm going to explain that on the next slide. Okay, so here's one reason why you might get a different story for a gene, for different genes. And so suppose that I have three species here, human, chimp, and gorilla. The fat tree, again, is their speciation history. Okay, so there is speciation there and a speciation down here. Now, what I show you inside of the branches here are sort of populations at different times, different generations. And so each of these blue dot is supposed to be an individual. And so this is one generation, this is a generation going back, and generation going back, and so forth. And now suppose that I take one gene, say the insulin gene, and I take one individual in humans, I can sort of track backwards in time where this particular gene came from ancestor to ancestor to ancestor to ancestor. And I can do the same thing for the same gene in an individual in chimp and gorilla. And if you go back in time, eventually these lineages are going to meet inside ancestral populations. And so this one eventually met with that one, this one eventually met with that one, and so forth. And that creates a tree. It's a tree structure that I'm going to call a gene tree, the red gene tree here. And one thing that you can see is that if you think of this as a rooted tree, it's actually a different tree from the fat one. Right here, on this particular gene, it looks like chimp is more closely related to gorilla. The most common rhesus ancestor is sort of lower than chimp to humans. And this somehow happened because in this first ancestral population here, where these two lineages could have met, they didn't meet, just by chance. These are just happening by chance if you sort of go backwards in time. And so you ended up with these two meeting first, and so forth. And so this is an example of what's called incomplete lineage sorting, and it creates this discordance between the sort of gene tree history and the population sort of history. And so why do we care specifically about the red tree? Because this is where our data lives, right? The mutations are occurring on the red tree. This is the thing that we can reconstruct from the data, whereas the fat tree is not something that we observe directly, at least not from this genetic data. And so people have been interested in kind of modeling this thing and to try to understand the effect that it has on phylogenetic reconstruction. It turns out that it's quite common. So in particular, in human chimp gorilla, there's a certain fraction of genes that indeed look like chimp is closer to gorilla and so forth. And this is one of the reasons why this might be the case. And so what you do is you just assume that this thing is happening at random. And so you have these two lineages here. At every generation back, they choose uniformly at random one of the ancestors. And so there's a certain probability that they meet. This is called a coalescent process. And if you take the limits and things, you get sort of a probability distribution for observing this particular gene tree. So you have a probability distribution over gene trees. You can write down formulas. These formulas will depend on the time between this speciation and that one, and on the population size, which is this n over here. And so the shorter the time here is, the more likely you'll see these two lineages not meeting and maybe having an inconsistent sort of topology. So this is the probability of observing each of those. And you get this sort of representation of this distribution over there, where you can see that the smaller this quantity is, so either the bigger this population is, which makes it harder for these to meet, or the smaller the time is, the more likely you'll see sort of different topologies for different genes. And so this is called the multi-species coalescent, where you can generalize this to any number of species. You're just doing coalescence inside of each of these populations and kind of stitching the lineages together. And the other assumption that biologists will make is that because of recombination, what we'll assume is that different genes are doing this sort of independently of each other. Okay, so now if you have a big genome, for each of your genes, or each of your locus, whatever genomic location you're using to do this reconstruction, each of your locus is just independently going to go through this process of coalescence and produce its own sort of gene tree. And so in particular now, if I'm only using one gene, I might get a tree that's actually inconsistent with the true speciation history, even if I have an infinite amount of data for it. Now, so how do you do, right? So because I'm getting now this data where different genes will have different trees, I can use my previous methods for each gene separately reconstruct a tree. The next thing is, how do I combine these trees together into something that gives me a consistent estimation of the species history, which is the bold tree that I want. And so it turns out that can be done in lots of different ways. This tree is actually identifiable from this information, so from the gene trees themselves. And there's some very simple things you can do. For instance, going back to this idea of breaking things up into small pieces, which is something that's used extensively in this, looking at quartets, so 4-species trees, it turns out that under this multi-species coalescent, if I restrict myself to 4 species, and I count across all of my genes how many times I've seen one or the other, the one that's the true one, the one that actually comes from the species history, is the one that's going to be the most frequent. Okay, so if I do this one quartet at a time, look at the most frequent ones, assuming that I have an infinite amount of data, that's going to give me the correct species tree out of it. In practice, of course, you don't have an infinite amount of data, and you also have errors in your reconstruction. And so the quartets that you're going to get are not necessarily going to be consistent with one species tree, but people have sort of very good sort of heuristic methods for doing kind of combinatorial optimization problem where they find a tree that sort of fits as many of these as possible or something like that. And these work very well in practice. One thing that's going to come up later is really the information that I'm using here is what I call unrooted gene tree topology. So I don't need to know the branch lengths or the root in order to do this procedure. I really need the unrooted topology of each of these trees, and that's enough to get identifiability of the species tree. And so here's where we are at this point, right? So again, just to summarize, we have this species tree that we are interested in reconstructing. Did I touch something? Okay. We have a species tree that we're trying to reconstruct, right? That's the sequence of speciation events that have led to the current species that are at the leaves of my tree. I don't get to observe that directly. For each gene in my data set, maybe the hemoglobin gene, the insulin gene, and myosin, and so forth, there's a gene tree, which is some random tree that was produced from this coalescence process. And then for each of these gene trees, what I'm actually observing is the genetic data that evolves separately on each of those. And so once I fix a gene tree, I can imagine that the substitutions have occurred on that tree. And so if you do the continuous time Markov process here, you get one of these tables. And so each of those is conditionally independent given the gene trees themselves. And now the goal is, from here, to recover the species tree. And what I've been showing you here is this sort of 2-step approach where I first, for each gene separately, I use that substitution table, the genetic data, to reconstruct their gene history. And then I combine all of these together into an estimate of the species tree by using, for example, this sort of quartet majority voting things that I explained. Does that make sense? Yes.
So if you take gene trees from genes that are closely linked on the chromosomes by linkage, and they're coming from the same individual, you should see a similarity in those particular gene trees. And unlike genes far away, they don't necessarily. Does that actually pan out when you analyze data?
So this is definitely going to be an issue. Right? So we've assumed here that our genes are independent, but of course that's not going to be true in reality. There's going to be some correlation between things that are close by. From the point of view of the identifiability of this tree, that's not necessarily a problem because as long as marginally each of these trees has sort of the right distribution, and so if I have enough of them, I'm going to be able to reconstruct my species tree. Where it's going to affect me is in order to get a good reconstruction, I need to have enough of these independent gene trees to be able to guarantee that I get the right thing out of it. If they're correlated, then it means that effectively I have fewer, in some sense, samples. So it will affect me, but not necessarily as badly as you might think that it would. Yes?
Right now, there are transformers and graph neural networks that could do something relatively similar So do you think that this could be compatible with those type of algorithms, or what would you see as the biggest difference between this approach and, for example, a transformer that is looking for sequences and sequence motifs?
Yeah, so it's very much an open question. People are definitely, of course, thinking about using deep learning in this particular field. And I've seen lots of papers about this. I've not seen any really kind of convincing method based on deep learning that can reconstruct trees. So this is not like and it's not like a classification sort of problem. You've got this massive space of trees, and so how do you turn deep learning approaches into something that will actually recover trees? I mean, there's ways of doing this. You can think of ways of doing this, but so far people have not really, I think, been super successful in this particular kind of application. But it's definitely something that people are thinking about. Okay, so here's my question. So here I showed you one very kind of natural setup. Was this the right thing to do? Okay, so should I really be building gene trees and building a species tree out of it, or should I be doing something else? Okay, so what would the something else be? It would be starting directly from the genetic data and trying to spit out a species tree out of it. And indeed, there are methods that do this. Okay, so far what I've shown you seems like a sort of natural thing to do, but what people actually do mostly in practice is this. They pretend that this is not a problem. Okay, so they'll just take their genome and just kind of concatenate it. So pretend that it's just stick the gene end to end into one big super gene, and then just apply maximum likelihood to build a tree out of it. You could certainly try to do that. You've got a lot more data, so you're going to get something probably that's much more accurate. What is the issue with doing this, though, is that it ignores the fact that different genes actually have different histories. It's not like the whole thing comes from one tree. You've got this sort of cloud of trees, if you want, or a mixture of trees that's sort of generating your data. And if you're applying maximum likelihood, pretending like it all comes from a single tree, then you have a sort of misspecified sort of maximum likelihood. And indeed, you can actually prove that this should not work. And so this is one theoretical results, some theoretical results of mine with some collaborators along those lines that say that it is possible to construct species trees where under the multi-species coalescent, if you apply maximum likelihood to the concatenated data, you're guaranteed to converge to the wrong thing. So one example is a tree that sort of looks like this, where I've got 6 species, where each of these branches is very, very short. And so in some sense, there's a high probability that none of these lineages meet, and they all meet instead inside of this big population at the top. And so what happens when that's the case? Then the coalescent process in that top population is going to do whatever it likes to do, not caring about what the structure of the tree was. And it turns out that the Kingman coalescent is what it's called, on a single population, likes certain trees more than others. It likes more balanced trees over unbalanced trees. And so it's going to spit out a tree that's unlikely to look like this one. And therefore, the most common kind of gene tree that you're going to see is actually going to be inconsistent with your tree. This is called the anomaly zone. And this is a bit different from what I said before. But when I talked about how to reconstruct trees, I said, well, if you restrict yourself to 4 species, the most common topology you're going to see is the real one. But that's only true for 4 species. Once you go to 5, 6, and so forth, you get cases like this where the most common gene tree topology is actually inconsistent with the species tree topology. And therefore, when you're going to do maximum likelihood, it's really not obvious here, but you have to prove that maximum likelihood is somehow going to tend to favor these trees that are more common in the data. Yes?
But you explained to us that you're actually not working with genes, but with just traces of DNA.
Yes.
So on some level, you already are doing this concatenation because there is no guarantees that a particular stretch of DNA will have a sort of the same mutation rate for all the bases. So maybe it's a question of what is the right scale over which you have to concatenate.
That's a very good question, yes. And that's a question of controversy and debate. In practice, if you want to apply one of these coalescent-based methods, you have to break things up into pieces that you know are going to be roughly the same trees and roughly the same parameters. And these might be short, and if they're short, it means that you're going to get bad inferences there and so forth. And so there's definitely a problem. And so people that criticize these coalescent-based methods will say exactly this, that how do you know that you're breaking things up in the right way? At the end of the day, maybe you're just doing concatenation anyway, so why not just do that? But as a theoretician, the fact that I can prove that it doesn't work bothers me. And so what else could you do? Well, I just told you that the problem with this concatenation approach is that I'm doing this reconstruction. I'm doing the maximum likelihood under the wrong model. Well, what you could do is do maximum likelihood under the correct model. You could say, well, let's model the whole thing. Right? We know how to do this. We have a species tree. We have a bunch of gene trees. We observe these. Again, under the assumption that we've broken things up in a way that you have regions that have the same kind of, that are not recombining within each of them. I could write down the likelihood of observing the data, right, and apply a proper maximum likelihood method to this. But as this arrow is trying to show, it's a very, very slow kind of thing. So people have implemented this, but it doesn't scale very well. Why does it not scale very well? Because if you just do maximum likelihood over one gene tree, one thing that works for you is that it's very easy to compute the likelihood of a data given a tree. There's a simple, what's called a pruning, or it's just like dynamic programming on a tree. And the fact that you can compute the likelihood relatively fast, if you're clever in how you search spatial over the space of trees, it seems to converge fairly quickly to the right thing. Whereas here, writing down the likelihood of a data means not just writing down the likelihood of each of these, but summing over all possible history inside of the species tree. And that thing is really very slow. And so in the end, it's sort of hard to apply these methods unless you have a small data set, in which case they actually work well. What else could you do? Well, it turns out that this previous idea also of using distance methods also worked to some extent here. I could concatenate my genes and then apply the juke scanner distance. And it's really not obvious why this should work. You have to prove it. But it turns out that it actually gives you a consistent method that actually recovers the tree. This is called METAL. And there are other methods that people have developed. One of them is called SVD quartets. That, again, sort of combines things across all of your genes. And then using some nice sort of algebraic properties of the matrix of the probability of different patterns. This is a very cool method. They can show how to reconstruct things, the tree correctly. So there's lots of different methods. Some of them work better than others in practice. So this is a very recent study. I don't know if it's even published yet. It looked at metal in particular, but about all of these methods. And it took a tree that's really hard to reconstruct, which is the sort of the tree of birds. There's lots of, I'll come back to why it's hard in a second. And of course, we don't actually know the truth, right? This is one issue with these problems. We don't actually know what the real tree of life is. You can do some simulations, but simulations are always sort of limited in different ways. But there's lots of other information that biologists have where they're pretty certain that some of these branchings are really there, and some of them are not. And so looking at this, I won't go to this table, but green is good. And so each column here are different methods, and each row here is a different branching. And green means that they got the right thing, and the other colors mean that they didn't. And what you can see is that the thing that really works the best is this concatenation with maximum likelihood approach. It gets pretty much everything correct. The other thing that works pretty well are this very first thing that I showed you, this two-step procedure where you build gene trees and then use quartets to recover the species tree. That also works very well in practice, but some of these other methods like SVD quartets and this sort of metal type thing actually does quite poorly, at least in this data set. So the question is, what can we say about, theoretically about these different methods? Why is it that some of them work better than others, some are more accurate than others? And how can you, from a theoretical point of view, kind of try to get a handle on these questions? Yes?
You're choosing a pool of genes that you want to use. What happens if
Yeah, so you get this sort of incomplete, right, I haven't showed you, but you get this incomplete data with some genes are present in some places, some genes are not present in other places. You can still reconstruct partial trees and combine them together, or you can just try to use only the ones that are kind of common across all of them. There's different approaches there in practice that are used. And these different, in this particular study, I can't remember exactly what they've done here. These are very good practical questions. So how do I compare different methods from a theoretical point of view? And in particular, can I prove anything about this question of, is this really the right thing to do, to build trees first and build the species tree? It seems that in practice, if I just ignore the problem completely, I get something better. I guess, what can I say about this? And that turns out to be a hard problem. How much time do I have? Maybe 10 minutes at most. And so I'm just going to tell you a little bit about, as a theoretician, how do we try to understand these questions of how do you compare the sort of statistical accuracy of different methods from a theoretical point of view. And so this is the tree of bird that I mentioned before. It is very hard to reconstruct, and there's a lot of controversy around some of these branchings. And you can sort of see why in this picture, right? So the time goes like this, and so this is a contemporary species. I mean, this is just like 50 of them. This is sort of the best estimate in some sense that we have. And then going back in time, you can see that something happened 65 million years ago. The dinosaurs went extinct, and then suddenly birds, there was this, what is it called, rapid radiation, adaptive radiation that happened, and suddenly tons and tons and tons of bird species came along. And so what that does here, this very, very fast branching, it creates these very short branches that are very deep inside the tree. And this is exactly the thing that's hard to reconstruct. Short branches are hard to reconstruct because intuitively, there's very few mutations along there. It leaves very little signal. And of course, the deeper things are, the harder they are because the mutations and then the noise will just kind of blur the signal over time, and so you're losing that information since you're only looking at what happens at current times. And so how do you turn that into an actual sort of theorem? Let's look at short branches. Right? So why are short branches? Go ahead.
If there are any species that were lost in those short branches, is it okay to, or can you reconstruct that?
So any kind of, you may be talking about extinction, maybe?
Yes, like a species that got extinct.
Yeah, so throughout this, right, it's assumed that the only thing that I get to reconstruct is what's called a reconstructed tree is the things that actually survive, right? Any kind of extinction I cannot tell you about, but it doesn't affect the rest of the model, right? If you think about how this model works, the fact that something goes extinct, that you just prune a branch from the tree, and the rest of the process just naturally goes along the rest, which is not necessarily the case for some of the other processes that I talk about, like lateral gene transfer or gene duplication and loss. Those, if you have extinctions, it can affect the process that you actually observe. But here, this particular process is sort of consistent. So very briefly, how do you prove that things are hard? This is where information sort of theoretic methods come in. If I have a short branch, okay, so F here is going to be the length of this branch. Think of it as the expected number of mutations along that branch on a given site. Then F is going to be this thing that's very small. And so to prove that something is going to be hard to reconstruct, I'm going to compare two different trees. And so this is A being sister with B. This is A being sister with C. Everything is the same. The branch lengths, everything else is the same. They both produce a distribution of data. And so let me just, for now, just focus on the first step of my process, just the substitution process that creates this genetic data on a gene tree. They create two different distributions. And what you can show is that unless you have what's over here, K is the number of columns in my data, so the number of sites that I have. Unless K scales like 1 over F squared, no method whatsoever is going to be able to sort of be able to tell these apart with high probability. And then the way you prove this is just by kind of computing a notion of distance between these two distributions. And that tells you something about the success of any kind of hypothesis testing method. Yes, I won't go into the details, but you can do these sorts of calculations. You can do the same thing for the second step. If I give you the gene trees that are correct, you also need a number of gene trees that scales like 1 over f squared in order to be able to distinguish between two species tree. And that both of these requirements are achieved by the methods that I presented. So both maximum likelihood and this kind of quartet majority voting thing achieves this best possible, quote unquote, up to constants sample complexity issue. Now, what's interesting here is that, so the proxy that I've sort of been using here in order to get a sense of how accurate something is how much data do I need to be guaranteed that this method will reconstruct the correct thing with high probability? Okay, so it's very hard to just, giving a method and a data set, what's the probability that it's going to do something correct? What kind of mistakes it's going to do? It's very hard to characterize this theoretically. The kind of, the trick that people have been using is how much data do I need to be guaranteed that eventually I'm going to get the right thing with high probability? And how does that depend on the parameters? And so here I showed that the shortest branch length in particular affects that sort of sample complexity. But here, in my kind of genomic scale kind of data, this particular proxy becomes a bit more complicated because there's two ways in which I can augment my data. I can have longer sequences, so the more sites per gene, but I can also have more genes. Of course, in practice, biologists have whatever they have, right? But in this sort of theoretical thought experiment, if I want to try to understand sort of the rate of convergence in some way of these methods, I have to think about how I can get more genes or I can get longer genes. And so what you have is this sort of two-dimensional picture of the number of genes. And just to simplify, I'm assuming that each gene has the same length here. Where can I reconstruct in this picture? Given a certain number of genes and a certain sequence length for each of them, where can I reconstruct and what methods actually achieve this? So you can do the same sort of information theoretic argument. You compare two of these models, and what you can show is this sort of thing. So we've been working on this for many years now, and we've been kind of developing this sort of phase diagram of where in this space is it possible to reconstruct species trees with high probability. One part of it, so this is log K, log M scale. One part that I've talked about so far is this part up here. If I'm able to reconstruct each, so if I have sequences that are long enough that I can reconstruct each gene tree separately with high probability, and I have enough of them, this is this two-step approach, then I know that I'm good. And these are these sort of gene tree topology-based methods. Vice versa, it turns out that there's this non-trivial trade-off between the number of genes and the length of the genes, where if you're down here, you cannot do anything. And that comes from this kind of information theoretic method of looking at the distance between these two distributions. And it has this weird kind of trade-off where it's sort of this slope -1, -2 or something here. And then we've been filling the rest of the gaps. It turns out that different methods work in different parts. I won't go through all of this. But we have various things. And so one thing that's, we started out doing this by trying to do this on very simple, with the sort of simplest setting, with the strongest sort of assumptions, and try to kind of relax these assumptions as much as possible. And one sort of natural question that arises here is, how complex do I really need these models to be to kind of capture the empirical behavior? At the end of the day, we want to be able to say something that says, well, yes, if you get a good theoretical result, hopefully that's going to reflect in practice. Because these are suggesting sort of new methods, and you'd like these new methods to work well in practice. And so one thing that we've encountered, one problem we've encountered with this is this question of, how far do we need to go? How complex do we need to make these models to reflect reality? I mean, already the stuff that I've shown you is this very complicated sort of processes. You can make them more complicated, and it turns out that some of these things matter more than others. And so one thing that we've done recently is to look at variable mutation rates across site. And so suppose that now each of my gene, instead of just going through this process, I also have this extra step where some genes are evolving faster than others. And so it might look something like this. Some genes are kind of being stretched by a random scaling factor. That turns out, you can do the same sort of argument, that turns out to have a big impact on this phase diagram. Once I include this in here, my nice sort of cute trade-off kind of disappears, and I get this much simpler picture now where you need to have a certain number of genes to be able to succeed, otherwise it doesn't. Now you still have parts of this diagram which we don't fully kind of understand. Down here, for instance, if you have short genes, but you have enough of them, can you still reconstruct, and what kind of methods are doing well there? I won't go into the sort of things that we've been doing since I'm out of time here, but one new method that sort of came out of this is this idea of kind of breaking up your data. So if I take a particular gene here, and so the fact that it has, I have multiple genes, each of them has a certain length, I can start doing more clever things where I can kind of break up my sites into two subgroups and being clever about how I construct estimators of these sort of distances or the 4-point condition that arise in this. Without going into the details, you can come up with methods that are kind of robust to the kind of variation across site that I was showing you, and might be good in practice. In fact, a very recent paper that just appeared in Science, independently from this, some other group that was at this ICERM program last semester came up with this method that uses very similar ideas and seems to be beating all of the other methods in practice. So there's still room for, my point is there's still room for coming up with new methods that are kind of inspired by these sort of theoretical considerations here. And so in the end, I did not really answer my sort of question, which was, is it a good thing to reconstruct gene trees or not? It was just a way to kind of introduce different methods. We don't really have a good answer for that. Some methods work better in practice than others. Some have better theoretical properties. And we're still kind of trying to figure out where the best of both worlds can be found. For the grad students among you, if you want to learn about some of the mathematical techniques that we use in this work, I've got this textbook that came out last year. And otherwise, thank you for your attention. Yes.
I just have a quick question. So, in all of the work you showed and in the methods, it seems like the trees are just about existing species that are still alive. What if I want to take seriously the hidden states, and I want to construct trees such that the extinction events are in the tree itself? I imagine you run into identifiability problems, but you can say things about the number of extinct species and things of that nature.
So you would have to start making lots of assumptions about how the tree can evolve. As I said, this particular class of model that I showed you, if you prune things, it doesn't change the distribution at the leaf, and so you learn absolutely nothing. There are other cases where I didn't talk about lateral gene transfer, but if you have lateral gene transfer, things might have left some signal, and you might maybe be able to reconstruct things like this. But I've not seen any methods that, as you said, it becomes an identifiability issue of what happens. Where is the signal of the things that disappeared is really not trivial. I think that was a question.
Use heterozygosity within a given species to put bounds on the incomplete lineage assortment, and also, in the same vein, why doesn't the mutation rate enter your formulas for the short branch estimates?
The mutation rate was hidden in the F here was really T times mu.
I see.
So
Okay,
it was
got
the
it. Okay.
time the mutation rate.
And so, but you must have data on heterozygosity within a given species. Shouldn't that put some bound on how bad the incomplete lineage assortment can be?
I don't know the answer to that. I'm sure that the biologists who do this would take that into account if they can. But I don't know about that. Yes.
Thinking about this quartet majority vote, and so like you have many gene trees, and some will side with the consensus, right, and some will be opposing the consensus.
Yeah.
What I was thinking is, like, as you said, like selection would distort these trees, like to a greater extent. So we could have independent measures of selection on these sequences, saying like how many substitutions are, like neutral when you're doing proteins versus not, like these kind of test of selections. Do we see like that the ones that are not siding with the consensus have an elevated signature of selection?
That's a good question. People have started looking at things like this. Selection specifically, there are different scenarios that can create certain trees to be wrong in a certain way. There's other issues also, is just the question of these short branches also mean that you make more estimation error when you build the gene trees. So there's lots of things that happen there. I don't know specifically about what you're asking. I don't think I've seen people doing this, but people definitely look at once they've reconstructed these trees, yes, the genes that have different histories, thinking about why that might have happened, and whether or not there are other things that they can learn from that. Yeah. Yes.
Have you tried to do these gene tree reconstructions from Y chromosomes? Because they're hemizygous. There's only one copy per genome. They're only in the male lineage.
Yeah, so I don't
I
know.
guess in birds it would be ZW.
Yeah, I don't know. I don't know whether that would be something useful. I'm not an expert on the empirical studies. What I do is the math. I read them because I want to try to do things that are relevant to them, but I'm not an expert on these sort of details about empirical studies. Yes.
Okay. One thing I noticed in your early slides, you were showing mitochondrial DNA, and to my understanding, I don't know, that's not on the chromosomes, so I don't think that has the homozygous heterozygous issue, so there might be something there. I had the question of whether you could do this with protein sequences of amino acids and use it for, say, orthologous proteins and abstract one level above. Would the theory still apply with the generalized notion of distance?
Yeah, so you could definitely define models of, say, substitutions on proteins. And from a mathematical point of view, at least, you can definitely define notions of distances and so forth. Selection might play more of a role in these, and how exactly that affects the reconstruction in practice, that I don't know. But people definitely build trees from protein sequences as well. Yes. Over here, yes.
That there's a clear probabilistic model going from the gene tree to the observed genome. But it's not entirely clear to me how well defined the species tree is, because my impression based on your presentation is that the genes can diverge either before the population diverges or it can diverge afterwards. So if you just see all these gene trees, to what extent do they really define an underlying population tree? And if there is a say probabilistic model taking you from the species tree to the gene tree, doesn't it also depend on things like the population size at every intermediate point?
So for your second point, yes, I swept a lot of things under the rug. When you define this multi-species coalescence, the population size definitely comes into play. And it was in my formula. There was an N in there, but the population size could change from different populations, or it could change over time. And people have defined multi-species coalescence accordingly. For your first question, the assumption here in the species tree is that once the population have split, there's no exchange at all. So you cannot have coalescence before going back in time, before the actual speciation event. So all of these coalescences are happening in ancestral population. You could have more complicated, of course, in population genetics, they have more complicated model, maybe where there's migration or introgression and so forth.
But doesn't it almost get at this philosophical question of what is a species? Like, isn't there a parameter that you can tune, and in one limit you end up with a species containing 5 individuals, another? Okay.
Yes, so as a mathematician, again, and my thing is just once they split, there's nothing in between.
All right.
Maybe last one, right? M0.
Thank you. I was wondering if you can weigh different regions of the genome differently, like transcription factors, protein coding, pseudogenes, during the concatenation step to see if the distances change? And would that be good?
That's a good question. One thing that people do in these summary methods is the genes, right? So they reconstruct a tree and they might have some bootstrap values on how how accurate they think that reconstruction is going to be. And there are methods that put weights on different genes that reflects how, how accurate they think they are. But the level of concatenation, I don't think I've see that as an interesting idea.
Thanks.
Okay, thanks to all of you.